My name is Sophia, and I am a PhD candidate in the Department of Chemical and Biological Engineering. Today, I'm going to be talking about aging and our quest towards understanding how and why this happens. As humans, we're all too familiar with the process of aging. As we grow older, there are obvious physical changes that happen to our body. For example, our hair starts to gray, our skin starts to wrinkle. We might not be as nimble or as spry as we used to be. Internally, we're also more susceptible to diseases such as Alzheimer's, heart disease, and even cancer. Additionally, aging also diminishes our internal immunity, which means that whenever we do get sick, it takes us longer and longer to recover. Overall, it's safe to say that aging is not really a desired process. However, to one day be able to curtail the progression of aging, we need to first understand what is happening to our bodies at a molecular level as we age. Our bodies are composed of organs, which then are composed of cells. Each cell in our body can function as an independent unit where each part of it will work together to help the cell stay alive and function properly. The most important part of the cell is its nucleus, which houses our DNA. This is all the information that the cell needs in order to survive and function. However, in order for a cell to use this information, it first needs to be processed. And the way that cells do this is it first transcribes this DNA into RNA and then translates this RNA into proteins. These proteins are the final products that the cells uses on a day-to-day basis. You can kind of think of this as a restaurant where there's a chef with a book of recipes, and in this book of recipes, there's directions on how to make all of the dishes that the restaurant serves. Here, the book is the DNA, and the final dishes that the restaurant serves to its patrons are the proteins that the cell uses on a day-to-day basis. However, the chef can't possibly make all of the dishes by himself, so he delegates all of the cooking to his sous chefs, and he just kind of oversees operations. The sous chef, however, doesn't really know how to cook any of the recipes, so in order for the sous chefs to make the proper dishes, the chef copies recipes out of his recipe book and gives it to the sous chef as directions. These directions are like the RNA molecule that is in your cells that is used to make our proteins. At any given time, there are a lot of different sous chefs in a restaurant making a variety of different recipes. Just as at a given time in a cell, there are a variety of different RNA molecules that is being used to create the proteins that the cell uses. And just as you can learn a lot about a restaurant based on the type of dishes it cooks and the type of recipes that the chef gives his sous chef, you can learn a lot about a cell based on the population of RNA molecules in a cell at a given time. For example, if you see that there's a cell that has a lot of RNA molecules that is used to create, say, proteins that transports oxygen, it's safe to assume that this cell is a red blood cell. This method of probing the RNA population in a cell at a given time is known as RNA sequencing, and it allows us to get a more detailed snapshot of the characteristics of a cell at a given time. As we age, the RNA populations in our cells will change. And you can kind of think of this, going back to our restaurant metaphor, like the different types of dishes that restaurants serve throughout the day. In the morning, the sous chef will make more breakfast-type foods like pancakes and eggs. And during lunch, the sous chef will start making sandwiches and salads. Finally, during dinner, he'll start making main course-type foods like steaks and ribs. By performing RNA sequencing on cells at different time points, we can, for the first time, trace cellular characteristics of a cell throughout time. And by expanding this analysis to a variety of different cells, we can now compare and contrast how the patterns of aging in different types of cells. This is extremely exciting because for the first time, we can start asking and answering questions such as, do all cells age in a similar manner? Do all cells age at a similar rate? We can look at a specific RNA molecule and see how aging affects it in a variety of different cells. And we can even maybe uncover underlying cellular mechanisms that govern aging in all cells. This will allow us to one day be able to pinpoint specific RNA populations that is integral for youth and vitality, so that one day down the future, we can design and engineer interventions so that we can live healthier for longer. Thank you.